Good afternoon, everybody. It's a pleasure to be able to be here at this meeting today and give this presentation on Enzyvant. It's really been a great collaborative meeting, and it's really in that spirit that I want to talk about and share some of the experiences that we've had getting our lead asset approved, Rethymic, going through the FDA approval process, and share some of the experiences that we've had to help benefit all in the community who are helping to develop these regenerative medicines. So at Enzyvant, we're focused on the development of regenerative therapies in rare diseases where there's a significant unmet medical need. I mentioned our lead asset is Rethymic, which was one of the 6 products that obtained their initial approval in the US, EU, or China last year. It's been approved for the treatment of pediatric patients with congenital athymia, one of the first RMAT designated products to be approved. Rethymic is indicated for immune reconstitution in pediatric patients with congenital athymia. It's not for treatment of patients with SCID, which is a distinct primary immunodeficiency disease. Rethymic is derived from human tissue, human thymus tissue, and is designed to regenerate thymic function in children that are missing the thymic function. These are children that are born with congenital athymia. They're born without a thymus. Importantly, it doesn't require any donor-recipient matching, so those are distinct donors relative to the recipients. We're fortunate to partner with an academic organization that had been developing this product over a period of many years, more than 25 years, in fact. So we had clinical trial data, very durable and robust data set with 10 trials over 25 years. And we obtained several regulatory designations, both from the FDA and EMA, and we'll talk a little bit more about how we use those during the development of the BLA and the review period. This is an overview of the process for manufacturing, treatment, and the mechanism of action of Rethymic. I mentioned that the thymic tissue is from unrelated donors. It's obtained from pediatric subjects who are undergoing a cardiac heart surgery. And for the surgeons to access the cardiac field, they frequently need to remove the thymus, and that's treated as medical waste at most universities or hospitals. But we've actually worked with our collaborators, and that thymus, when it's removed, can be used as our source material for the generation of Rethymic. So that donated thymic tissue is processed over a 12 to 21 day manufacturing period that's important for depleting the T cells in that donor thymus. And it is then used, it's implanted actually, it's sliced and implanted into the quadriceps of a recipient child in the surgical procedure. So that's the recipient child with congenital athymia. And the mechanism of action in vivo has the migration of the T cell progenitors from the bone marrow to those implanted Rethymic slices. So it's essentially functioning as a thymus, but in the quadricep of the recipient child. And it's in there where they develop into immunocompetent recipient T cells, so naive CD4 or CD8 cells. This is the Kaplan-Meier survival curve that's in our US package insert, obtaining the FDA approval. What you can see is, looking at the left-hand side, is a natural history population. So these are subjects that did not receive treatment with Rethymic, and you can see that the condition is uniformly fatal by 3 years of age. For those subjects who did receive treatment with Rethymic, you can see that there is some deaths occurring in the early period, primarily in the first year, and then we have almost essentially a flat line after that. We have 77% estimated survival at 1 year and 76 at 2 years. So those that have survived to 1 year, we have a 94% estimated survival with an average follow-up of over 10 years. So obviously a robust data set. And the reason we have some deaths occurring in that first year, because it takes time for the immune system to reconstitute. It typically develops about 6 to 12 months after treatment. And beyond that period of time, these patients have really a pretty strong, relatively normal immune system to provide that survival benefit. So there was 105 patients in the safety database, and there were 29 patients that died, 23 of those deaths in the first year. The most common causes of death were similar to those of the underlying condition of congenital athymia, so infection, respiratory failure, hemorrhage-related events, and cardiorespiratory. There were no deaths considered related to the treatment after a year. And again, with the common AEs are also representative of the underlying condition. Approximately 76% of the subjects experienced an AE within 2 years of treatment. So this is what's shown in the label, with the most common AEs being hypertension, cytokine release syndrome, hypomagnesemia, rash, renal impairment, and graft versus host disease. I want to transition now and talk a little bit about some of the approaches that we've used at Enzyvant that I think can be more broadly applicable to all of us who are working to develop the regenerative medicines. You can tell that this is a unique therapy, as are so many of the therapies that we're working on developing. So we've had to overcome a variety of regulatory and CMC hurdles, as well as working with a single site of administration and treatment for the commercial product, so creating new business and market access models as well. And really, there's a lot of novelty with this program, but there are certainly some shared experiences that can benefit all of us in the field. Within the regulatory field, I've been fortunate to be the regulatory lead over the last really 5.5 years now, since we initially in-licensed the product. And there was a different set of thinking that went into it. We really took some time in the beginning to evaluate the data that we had, and took time to obtain various regulatory designations. It was the first product to obtain both the RMAT and breakthrough designations. Ultimately obtained the rare pediatric disease designation, which enabled receipt of a priority review voucher upon approval, as well as we have orphan drug designations, both US and EU, and in the EU, the Advanced Therapies Medicinal Product designation. And I'm going to talk a little bit more about how we use those designations during the review and approval process. These designations helped us, have significant interactions with the agency on a more timely basis. I'm going to highlight that, during COVID, I think the designations were important just to maintain a standard pace of interactions with the agency and ultimately obtain our approval on the PDUFA date. With regards to the CMC work, there was a lot of distinct manufacturing and specialized approaches with this product. We did obtain a complete response letter in December of 2019, and we've pulled together a team that was able to successfully remediate that in less than 2 years. So this was a CMC-focused CRL that was related to the manufacturing process that occurs at a partner in an academic partner, so an academic CRO, contract manufacturing organization. Ultimately, the approval was the result of this BLA resubmission that we pulled together and submitted in April of 2021. The summary basis of approval is available publicly, and you can look at the number of distinctive approaches that we took and how we addressed the various aspects of the complete response letter and the skills that we developed and applied here that were unique. I think one of the other aspects that the skills that we've developed at Enzyvant is partnering with academic organization and doing that effectively. We all know that academia is a tremendous source of innovation, but their development is not necessarily targeted with FDA approvals or health authority approvals in mind. And so beyond that, it's important to work with our academic partners and think about our shared and mutual goals, which of course is the ability to treat patients. And always keeping that shared objective in mind, we're able to reach mutual agreement and understanding, prioritization, resourcing to support efficient development. I can say that with the clinical database, for example, it's a significant clinical database. Mentioned there were 10 clinical trials for over 100 years, over 25 years. The clinical data, existed in 3-ring binders and a large number of cabinets, and converting all of that into an electronic database that would be suitable for an FDA submission and review, going through the inspection process is a significant effort. So, and we also, of course, we're partnering closely with the academic organization that is our site of administration for the product. So there are a lot of commercial interactions. Our Chief Commercial Officer, Johanna Russell, has done a great job with that. She's presented on a panel yesterday. Certainly taking novel approaches, ensuring we have an appropriate level of risk sharing so that we're all on the same page and can proceed with patient treatment. We have a great leadership team at Enzyvant. Several of the leaders shown here, our CEO is Bill Simmons, Johanna is here presenting, and Jim Luderman is very heavily involved in all of our early development and partnering efforts. I also want to mention that one of the benefits that we've had at Enzyvant and continue to leverage is our global resources. We're a relatively small company, less than 50 employees, but we do have the benefit of being a subsidiary of Enzyvant Biopharma, which is a wholly owned subsidiary of Sumitomo Pharma. So we're based at dual offices in the US, in Cambridge, Mass, and Durham, North Carolina. And Enzyvant Biopharma, as I mentioned, it's part of Sumitomo. And Sumitomo has a significant effort. They're based in Japan in regenerative and cellular medicines. And we've partnered closely with their group throughout the development of the BLA and the review and approval process. So thank everybody for listening today, and look forward to interacting with you all through the rest of the conference.